The overall goal of this procedure is to selectively label active phagocytic cells, which clear out cell corpses in stroke. This is accomplished by first preparing components for the tyramide-based signal amplification reaction. The second step is to prepare the sections. Next, the labeling reaction is performed using the enzyme Vaccinia topoisomerase and ligatable fluorescent probes. The final step is tyramide-based fluorescence signal enhancement. Ultimately, fluorescence microscopy is used to assess phagocytic clearance of dead cells in tissue sections of focal brain ischemia.
The clearance of dead cells by phagocytes plays an important role in brain recovery after ischemia. The presented method determines the efficiency of this process by labeling only those phagocytes which actively remove dead cells.
Though this method provides insight into the ischemic brain, it can also be used to study ischemic injury in other organs and tissues where phagocytic clearance of dead cells is important.
Although the tyramide signal amplification, or TSA, step occurs in the final stage of labeling, preparing for this reaction can take more than an hour. It is therefore more convenient to make the TSA reagents first. The first step is to make the fluoro-4-tyramide stock solution. Add 300 microliters of DMSO to the tyramide reagent vials included in the TSA labeling kit. Next, prepare the TNT wash buffer. To completely dissolve the blocking reagent when making